Hi, my name is TJ Odom, and I'm a third-year PhD student here at the University of Georgia at the Odom School of Ecology. Climate change is one of the greatest present threats to global biodiversity and human health. Many organisms are having to shift where they occur in the landscape to track optimum habitat.
When we think about which species might be sensitive to environmental change, salamanders certainly come to mind. In fact, here in the southern Appalachians, several salamander species are currently experiencing shifts in their elevational distributions in response to changing climate, moving up in elevation to track cooler temperatures and down in search of warmer conditions. This is called climate tracking, and it's a strategy widely used by both animals and plant populations given enough time. Alterations in the distribution of wildlife across the landscape can lead to the overlap of newly climate tracking species, and when these host species overlap, if they're closely enough related, they can hybridize. Plethodon shermani, a high-elevation salamander species, and Plethodon teahali, more commonly found at lower elevations, are two species hybridizing along elevational clines where their ranges overlap. Here in the southeastern Appalachians, this incredibly dynamic hybrid zone is widening, with hybrids of P. teahali and P. shermani encroaching on high-elevation populations of P. shermani. Hybridization in wildlife can have some crazy outcomes, but one common result is resistance or tolerance to infection from parasitic diseases. Preliminary surveys of parasite communities in these salamanders found that individuals collected from populations of pure P. teahali and populations of hybrids contained fewer parasites than those collected from populations of P. shermani. This observation begs the question, to what extent does parasitism influence the spatial distribution and population dynamics of these salamander species and their hybrids? To ask this question, I'm combining contemporary field sampling efforts with surveys of salamanders from the natural history collections here at the University of Georgia, going back all the way to 1943, documenting parasite diversity and abundance in salamanders from sites containing pure parent taxa, as well as sites containing hybrids. If parasites are mediating competition between these hybrids and P. shermani, I expect to see greater parasite burdens in P. shermani when compared to the hybrids. Results of this work will inform understanding of the roles that parasites play in their communities as mediators of competition and potential drivers of movement for range-shifting hosts, and will highlight the utility of museum natural history collections for studying host-parasite interactions across a multi-decadal time span.